Hello everybody. Today we're going to be talking about development and plasticity in the brain. So plasticity is the idea that the brain is constantly changing throughout the lifetime, and the brain is changing. Why is it changing? Well, it's due to both experience with the world that's causing synapses to be created and different connections to be strengthened or weakened, as well as physical maturation that the brain develops as we develop. There's rapid brain development, especially early in life, that leads to the amazing increase in abilities that we see from infancy into childhood. So things like the prefrontal cortex is developing. The prefrontal cortex is really important for higher level thinking and cognition, and leads to the ability of even young infants to understand the idea of object permanence. Here's a picture. Oh, look at that cute baby. When infants are quite young, before the age of 9 months old, if they're looking at a stuffed animal or a toy or something that they're interested in, it catches their attention. But before they have developed object permanence, which again is one of the functions of one of the many functions, that's the prefrontal cortex, when they can no longer see that object of interest, it's as if it doesn't exist. Out of sight, out of mind. It's gone. And then they're on to something else. However, about 9 months old, as the brain develops and the prefrontal cortex develops, children develop the idea of object permanence, where if they're interested in a particular object, they like this little blue stuffed animal, and then you put something up in front of them so they can't see it, the baby will know that that stuffed animal is still there, and you'll see the baby maybe reach for around the paper to try to get the stuffed animal, maybe cry, maybe get angry because they know that animal is still there. This is an idea, just one of many, of how the brain changes over time, right? Think about all of the language skills and mathematics skills and all sorts of skills, motor functioning that increases over time. Well, all of that happens because of brain development. So we're going to watch a little bit about the beginnings of the formation of the central nervous system. We'll take a quick watch of this video.
Development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, and then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days.
So
at this stage, then, I'd like to draw your
the blastocyst, the ball of many cells.
Attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal, and the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the 3 beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye. And then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
All right. So, let's talk about the central nervous system. Remember, that's the brain and spinal cord, and prenatal development that you just watched. So the human central nervous system begins to form at approximately 2 weeks. That's very early on. That's often before a woman even knows she's pregnant. So that starts happening very early in development. At day 4, you end up with this blastocyst. This is that ball of cells, right, that was rapidly dividing and becomes a ball of cells, or a blastocyst. By day 18 of the pregnancy, the beginning of the central nervous system starts to form. The process of forming the neural tube, and the neural tube will become the brain and spinal cord, is called neurulation. So neurulation, remember that blastocyst kind of flattens out and then curves upward. So the dorsal surface thickens. The dorsal surface is the back, right, towards the back. The neural tube forms, and it's filled with fluid, right? That what will be cerebrospinal fluid. The forward end of the neural tube will become the brain. The rest of the neural tube will become the spinal cord. And like I said, that fluid-filled cavity will become the central canal and ventricles, and the fluid is the cerebrospinal fluid. And this all starts very, very early in development. So here's that formation of the neural tube. The cells flatten out, then kind of curve up on top of each other. There's that neural tube. The forward end becomes the future brain, and the rest becomes the spinal cord, with that fluid-filled center with cerebrospinal fluid. And here's just another video, or another picture of how this brain develops over time. And you can see at 3 weeks, you've got the hindbrain. The midbrain's actually quite large and a pretty small forebrain. By 7 weeks, here's the hindbrain again, midbrain still large. Forebrain is starting to form. At 11 weeks, again, this is very, very early in development. Look how much of the brain is formed. The forebrain is starting to get quite large and outsize the midbrain. And then at birth, you've got this very large forebrain, this small midbrain, and then the hindbrain. At birth, the human brain weighs about 350 grams. At 1 year, the brain weighs 1,000 grams, and the adult weight is about 1,200 to 1,400 grams. So you can see quite a lot of increase in weight of the brain between birth and 1 year old. The question is, what's happening there? And we will discuss that in a minute. So before birth, prenatally, so prenatal development, what are the stages of brain development? We start with proliferation, and that's the production of new cells. We have lots of new cells being formed early in prenatal development. The second part of brain development is migration, and that's happening right here, where neurons actually move toward their specific destination in the brain. Are they going to be part of the forebrain, or part of the midbrain, or part of the amygdala? These cells move to their appropriate area, and they're actually guided along by the glia cells that we talked about. Those support cells sort of provide a pathway for them, for the neurons to get to their appropriate place in the brain. And then once they're in the place in the brain where they're meant to be, differentiation is the next step, and that's when these neurons begin to form their distinctive neuron shape in terms of developing dendrites and that long axon. Now, after birth, postnatal development, what's happening in the brain, and this is that first year that we were talking about, where the brain grows so much in weight. Well, what's happening? It's not that there's neurons that are being formed. That all happens prenatally. But postnatally, the next step would be what we call synaptogenesis, and that's an increase of synaptic and dendritic density. The neurons that are there are forming lots and lots of new connections. It's a proliferation of connections between the neurons in the brain. In fact, infants develop more synapses, more connections, more communication than the human will need. And so at first you get this proliferation of synapses, and then based on which synapses are being used, which connections are being used and which are not, the brain goes through a process of synaptic retraction or pruning of synapses, meaning the synapses and connections that aren't being used die off. And that's supposed to happen. Those connections sort of go away. So you end up with a much more streamlined brain where the connections that are being used stay and the ones that aren't are pruned away. And eventually we have what's called programmed cell death. This is not a problem. This is a normal part of brain development, that you have any of the cells that are not being used and not important in terms of the over-proliferation, those cells will die off. All right, and then once you have a stable number of cells and the synapses are in place, myelination begins. And myelination, remember, is that covering over the axons of many of the neurons that helps with speeding up communication. And myelination starts at the spinal cord, then in the hindbrain, midbrain, and forebrain, so sort of from the back of the brain forward, and continues for many years. In fact, this process takes quite a bit of time, and this myelination process also leads to some of the weight gain in the brain. So it's those synaptogenesis, those synapses that are increasing, the dendritic branches that are increasing, the myelin that's increasing. That's what's causing the increase in brain weight, not so much extra neurons that are being produced. Now, what determines whether a neuron is going to survive or not? When a neuron forms a synapse, and we're going to talk about just neurons that form synapses on muscles, so motor neurons, the muscle produces something called a neurotrophin, which is basically food for the neuron, called nerve growth factor. And basically, if the neuron doesn't get enough nerve growth factor, it dies. That's the idea of programmed cell death. We also call that apoptosis. And so neurons really have activity-dependent survival. Without contacting an appropriate cell, the neuron will die. So if the neuron synapses on a muscle and that muscle is being used and that synapse is being used, that muscle produces nerve growth factor, and that is what the neuron needs to live. But if the neuron synapses on a muscle that isn't being used, or that connection isn't being used, there's no nerve growth factor for the neuron, and the neuron will die. And so that's the idea of neural Darwinism, the selection process of neural connections, that we initially form many more connections than we need, and only the most successful connections survive. How do neurons find their way to the correct location? Interestingly, they seem to follow a chemical gradient. And the research on this was done by Sperry and the severed optic nerve of a newt. So here we've got this newt, and there's an optic nerve that goes from the eye to the brain. And you can see the connections there. And in this research, they severed the optic nerve. So the optic nerve was cut. And even once the optic nerve was severed, the neurons would grow, the nerves would grow back and connect correctly. They did the experiment again where they cut the optic nerve and actually rotated the eye 180 degrees. So you can see that what was on the ventral side is now on the dorsal side. This blue neuron is now at the top. The red neuron is now down at the bottom. So rotated the eye, and still those neurons connected appropriately from one area, from the eye in this case, to part of the midbrain. And so research shows that these neurons connect and find their way back following a chemical gradient. In terms of new neurons, it was previously thought that there were no new neurons that were ever developed. You were sort of born with the neurons you have, and any neuron you lost, you were left with one less neuron. And every time you lost a neuron, it wasn't very good for you. And that is true. However, recent research suggests that some new neurons are produced, that we produce some new neurons after birth in the hippocampus, as well, and that's important for memory, and in the olfactory bulb, which has to do with scent. And so we still need more information. There certainly are not a lot of new neurons that are formed after birth, but we are finding that there are some, which is pretty interesting and exciting research, particularly if we think about recovery from brain damage. How can we get the brain to produce more new neurons? That could be a great intervention. In terms of which neurons survive, experience counts too. Experience helps determine which neurons survive. They've done a lot of research on animals brought up in enriched environments versus deprived environments. And so some of this research on rats in an enriched environment. So these are rats that were living in very fancy rat cages, right? With wheels and toys and mazes and all sorts of very cool rat things. And animals or rats that grow up in this enriched environment show a thicker cortex. More neurons survive. There are more synapses created, and they have improved performance on learning tests. And if you have compare this to rats that are raised in a deprived environment, so that would be a very plain cage with no toys, with no wheel, with nothing to do. Those rats have smaller, thinner cortices and do poorer on learning tests. So enrichment or deprivation effects are more prominently early on. We see the effects on the brain at an early age, more so than at a later age. And so it's really important, I think of this as a pediatric neuropsychologist, for infants to be stimulated, cognitively stimulated, have their environment be a very enriching environment. Now, by enriching environment, I don't mean expensive, like you don't have to buy all the most fancy toys, but have things around for the child to look at, pictures on the wall, talk to the child, music, play music for the child, different things like that. For my daughters, I hung from the ceiling right above their, well, not right above their crib, but in the middle of the ceiling, I hung little stuffed animals from the ceiling. So when they're looking up, they have something to look at. They're different colors and shapes. those are the kinds of things that can lead to optimal brain development. In terms of deprivation early on, those effects are also quite significant. They've done studies, so either on a deprived environment or with kittens. They sutured one eye shut for baby kittens for the first 4 to 6 weeks. And what happens is, because that kitten isn't getting any input from that eye because it's sutured shut, those neurons aren't sending signals, they're not getting any neurotrophin, right? And those synapses are basically unused. And so those neurons die off. And when you unsuture the eye, the kitten is blind in that eye. Because of the early deprivation, those neurons basically go through programmed cell death, and the kitten is blind in that eye. Here's an example of a stimulating environment on neural branching in a jewelfish, reared in isolation. That's over here. A has fewer branches, fewer dendrites, and then here in a fish raised with others, you can see has many more branches, more connections, and you can see that right at the neural level. And here are brain correlates of extensive music practice. So again, as you're enriching the environment, doing things that are cognitively stimulating, we see increases of gray matter, so that's the cortex, in certain areas of the brain. Again, just showing that cognitive stimulation leads to more connections, kind of a richer, thicker cortex. The vulnerable developing brain. What can go wrong? The early stages of brain development are critical for normal development later in life, and there are many things that can go wrong. We can have some kind of mutation in the gene, a genetic defect that can affect the brain. Exposure to toxins. If mom drinks alcohol during pregnancy, that can lead to a negative effect on the brain, a fetal alcohol syndrome. Infections can affect the brain, malnutrition, trauma. So there are many things that can happen during development that can negatively impact the brain. We see in a study of children of mothers who use cocaine during pregnancy, those children have a decrease in their language skills and a slight decrease in IQ scores, as well as impaired hearing. Children of mothers who smoke during pregnancy, that is correlated with low birth weight of the baby, correlated with sudden infant death syndrome, with attention deficit hyperactivity disorder, and with long-term intellectual deficits and impairments of the immune system. So these are what we call teratogens. These are substances that can negatively impact the developing fetus, and in these cases, the brain. Another potential cause of brain damage would be what we call a closed head injury. This is sudden trauma that doesn't puncture the brain. So getting in a car accident, a fall, an assault of some sort that impacts the brain. And you end up with, again, the brain is kind of floating in cerebral spinal fluid in this very bony skull. And so if you get a very hard hit on the head, whether it's a car accident and the head hits the seat in front, or a fall off of a high, fall out of a tree or something and hit your head, the brain that's floating in the skull basically rotates a little bit, kind of bounces around. We saw that the skull is very bony, and so the brain can be we call it a shearing injury, which means that your brain kind of rotates inside your skull and you can have damage all along the outside of the cortex. And you get two kinds of injuries often with a closed head injury. We call it a coup and counter coup injury. The coup is exactly where the injury takes place. So in this case, looks like somebody is punching the person in the frontal lobe here. And so that would be the coup injury right where the injury took place, or if somebody fell on the front of their head or something like that. But after that, the head and sort of bounces back, the brain sort of bounces back and hits the inside of the skull in the opposite direction. And this is the contra-coup injury. So you get another injury kind of on the opposite side of the brain. You can get this from repeated blows to the brain, concussions, etc. And closed head injury is one of the main causes of brain injury in young adults. Another cause of brain injury is stroke or a cerebral vascular accident, and this leads to is when you have a loss of normal blood flow to a brain area. Stroke is a common cause of brain damage in the elderly, although it can happen to children and even in infancy. In fact, that's a lot of my research is on very, very early strokes, so children who have had strokes either prenatally before they're born or within the first 6 months of life. That's much more rare. And you can see pictures of the brain here of a person who died immediately after a stroke, and you can see there's a lot of swelling. There should be a ventricle here, a space, but the whole brain area is swollen. And just like you would if you kind of hit your elbow or hit your leg against something, you end up with a bruise and a swelling. The same thing happens in the brain. Here's the brain of a person who survived for a long time after stroke. You can see that there is a greater size of the ventricle on the left side here, indicating some damage where cells were lost. And then this is a person who suffered a gunshot wound through the brain and died immediately. So there are two causes of stroke. In each case, blood does not get to certain areas of the brain, causing those areas of the brain to die. But there are two potential causes. The first is called an ischemia or an ischemic stroke. This is the most common and results from a blood clot or an obstruction of an artery. So if you have a blood clot in an artery that stops the blood from being able to flow through to the rest of the brain that was fed by that artery is not able to get oxygen and will die. There's also a hemorrhagic stroke. This is less frequent, but this results from a ruptured artery, a burst artery in the brain. And in this case, again, because the vessel bursts, whatever part of the brain was fed by that blood vessel is no longer able to get blood through. And in fact, neurons are around that area are flooded with excess blood, excess calcium, excess oxygen, etc. So not only do you lose neurons because those neurons are no longer fed by the blood because of either the ischemia or the hemorrhage, these strokes also cause edema. That's an accumulation of fluid in the brain, and it increases pressure on the brain, right? As your brain is getting more swollen, imagine that, it's getting more swollen, and your skull is very hard and very bony and doesn't stretch, and your brain is getting bigger, right, because it's swelling up, and that can lead to the probability of further strokes and further brain damage as your brain is swelling inside your skull. Also, we see a slowing of the sodium-potassium pump. Remember, the sodium-potassium pump is essential in keeping the right balance of ions in the intracellular and extracellular fluid for the action potentials to occur normally. And so you end up with the accumulation of potassium inside the neurons and a release of excess glutamate. Glutamate is an excitatory neurotransmitter, which ends up overstimulating neurons, and you get an excess of sodium entering neurons, and then the neuron dies, basically due to, even though the sodium-potassium pump is slowing, it's kind of an excess of or an overstimulation of the neuron that causes cell death. So either with ischemia, where you have a decrease in blood flow, or hemorrhage, where you have an excess of blood flow, this disrupts the blood-brain barrier. You get the edema or the swelling, and the sodium-potassium pump slows. All of this causes sodium, calcium, and other ions to accumulate inside neurons. Also causes neurons to release more glutamate, which is an excitatory neurotransmitter, basically overstimulating the cells, and eventually leads to cell death. How can we reduce harm from a stroke? If you have an ischemic stroke, a blood clot, there's a medication called TPA that is able to break up the blood clot. And so if you're able to get to a hospital very early on, they can administer this medication. The blood clot can hopefully be broken up. The blood flow is restored, and we can limit the amount of damage done by the stroke. So there's a lot of public service announcements and education on stroke. You might have even seen some announcements on television about warning signs of stroke and getting to the hospital immediately. Now, this only works for ischemic strokes where there's a blood clot, right? Not for a hemorrhagic stroke. Also, we can try to save cells in the penumbra, which is the surrounding area of where the stroke was. So if the stroke was here, this is the area of greatest damage, and these cells are perhaps going to die no matter what. But we can try to save the cells in the surrounding region. How can we do that? The most effective method seems to be to cool the brain. A cooled brain has less activity and lower energy needs, and there's less of a risk of that overstimulation that leads to cell death. And cooling the brain and cooling the person down can improve survival and behavioral functioning. This is kind of the opposite of what was done prior to this research. Prior to this research, people thought after you had a stroke, keep you warm, right? But now we realize it's actually needing to keep the person cooler to kind of lower the temperature of the brain, to decrease the stimulation in the brain, and to hopefully save some of the surrounding cells, because you will end up with less behavioral, potentially, consequences the fewer cells that die from the stroke. In terms of the effect of age on recovery, recovery is generally better if brain damage occurs at a younger age. Again, this is my research on children who have had strokes before 6 months of life, and what we find is that they do amazingly well. The brain is able to reorganize, and children are left with much more minor impairments than an adult who would have a similar size stroke. They've done research with rats with amygdala damage, and younger rats recover quicker. And as I said, you see the same with children. That a 2-year-old child with left cerebral damage will develop some speech, but an adult would not recover much language at all. Language is typically housed in the left hemisphere, and I see that with my research, that if a child has a left hemisphere stroke, every single child we see develops language, even if they're missing almost all of their left hemisphere. Whereas with an adult who has a left hemisphere stroke, you very often see language problems and the loss of language. In terms of brain damage, damaged axons do grow back under certain conditions. If an axon in the peripheral nervous system is crushed, it follows its myelin sheath. Remember, the myelin is made by Schwann cells in the peripheral nervous system, and they can get back to their target grow back at a rate of about 1 millimeter a day. But damage in the spinal cord and brain is relatively permanent. There's scar tissue that's formed by the glial cells, those oligodendrocytes, that makes a mechanical barrier to the axon regrowing. And the myelin in the CNS also releases proteins that inhibit axon growth. So really, those oligodendrocytes are very unhelpful for brain damage in the central nervous system. So instead of having axons regrow, what we do sometimes see are collateral sprouts, which are new branches formed by other non-damaged axons that attach to vacant receptors. So you can see here, if we have one axon, and here's the presynaptic terminal, or the end of axon 1, and here's the end of axon 2, and those are synapsing on the dendrites of another axon. And then this neuron is injured and dies, axon 2. You can see this collateral sprout, this extra sprout, these extra connections that grow to sort of fill in the space left by the damaged axon. So over several months, the sprouts fill in most of the vacated synapses and can be useful in some cases, can help the person recover some function. They might be worthless, or they could actually be harmful. Another way the brain compensates for decreased input and restores normal functioning is denervation or dissensitivity. Which is after the destruction or inactivity of an incoming neuron, the postsynaptic cell becomes more sensitive to a neurotransmitter. So the remaining neurons also increase the release of the neurotransmitter, and this can lead to recovery of near normal functions. So again, remember, if we've got two axons here that are both synapsing, or two neurons that are both synapsing on the dendrite of this cell, let's say they're both releasing glutamate, which is an excitatory neurotransmitter, and then this neuron is injured and dies, and so you're only getting glutamate from this one neuron. Imagine there's no collateral sprouting in this case. Well, what we find is that the postsynaptic cell can become more sensitive. That's the idea of supersensitivity, right? This postsynaptic cell can become more sensitive to a neurotransmitter, meaning it needs less of it in order to fire an action potential. And at the same time, this neuron that's left may start to increase the release of the neurotransmitter. And so that might lead to recovery of function. Plasticity after brain damage, phantom limb. This is the continual sensation of an amputated body part. It's very frustrating and sometimes can be extremely painful for patients, and I did some work in this area too as an undergraduate. Actually, it was the first research I ever did, and patients would describe if they had their arm amputated, patients with a phantom limb would feel as though their arm was still there, and sometimes it would be very painful. They'll say, my arm or my fingers feel like they're just squeezing very hard into a ball. I can't unclench my hand. My muscles feel very tight, but again, there's no arm there. So it's very hard to treat phantom pain. Why does this happen? Because we believe the cortex reorganizes itself after amputation of a body part, because it's no longer getting a signal from whatever part of the body was amputated. So in this case, here's our somatosensory cortex. And this part of the somatosensory cortex receives information about feeling from the hand. And so this is without an amputation. After, in this case, one of the fingers is amputated, what you can see here is finger 1, finger 2, finger 3, finger 4, finger 5. This is the sensory input. When the third digit is amputated, you can see that digit 2 and digit 4 kind of fill in that space, right? And so the original axons that were there degenerate, leaving vacant spaces into which other axons sprout. And so it may be that now when you get some stimulation on digit 4, that might actually, because those axons have innervated the space that was for the third finger, the person, or this case animal, would potentially feel as though they're getting sensory information on their third finger, even though it's not there, because these other neurons can kind of fill in the space. All right, thank you very much.